Ali, lecturer of medical biochemistry and molecular biology, faculty of medicine, Helwan University, Egypt. So our topic today is going, let me share the screen first. Okay, I hope it's shared already.
Yes.
No? Okay, so today I chose the topic of molecular basis of cancer. Why did I choose this topic? Actually, I chose this topic because cancer is considered the second most common cause of death after cardiovascular disorders in many countries. Approximately 6 to 7 million people die around the world from cancer each year. So I think that every medical student, every medical graduate should have this genetic background, especially that whatever his specialty will be, he's probably going to encounter a cancer patient. He should have some basic knowledge about the genetic basis of cancer, since cancer is a paradigm of acquired genetic disorders. So cancer is basically a genetic disease. And with the revolution of knowledge, which has been going on the last few decades, with the whole genome sequencing, development of tools to isolate, to identify genes, proteins, which may be involved in carcinogenesis, all of this revolution of knowledge aided in reaching an understanding of the malignant cell, which basically will facilitate the diagnosis and treatment strategies. So any oncologist cannot spare this knowledge, because it's going to help him so much in his medical practice. And as medical students, I hope that this lecture will be of benefit to all of you. So let's first navigate to the main objectives I'm going to cover in this lecture. First, we are going to talk about just the features of a cancer cell in short. And then we are going to see the genetic damage predisposing to carcinogenesis. What are the cancer genes involved in this process? We are going to explain the role of microRNAs in cancer. Then we are going to mention the role of growth factors. We are going to point out to the tumor microenvironment and the immune response shortly. Then we can explain how a cancer cell evades apoptosis, identify the changes in telomerase activity in a cancer cell. And finally, we are going to outline how understanding a cancer cell biology led to development of new therapeutic strategies. So let's start. Like I said before, that the genetic damage is the main initiating event in carcinogenesis. Cancer is a genetic disorder. So there are principally 4 classes of genes, which are, when affected by such genetic damage, they can result in development of a tumor. Many genes we have inside our body that when they are apparently mutated, they can result in carcinogenesis. These mainly are proto-oncogenes, tumor suppressor genes, genes involved in DNA repair, and those involved in apoptosis. Mutation in any of these genes, genetic damage in any of these genes can lead or can initiate carcinogenesis. So a normal cell can turn to a cancer cell. For example, this slide presents that exposure to a physical damage can activate a proto-oncogene into an activated oncogene, which can turn a normal cell to a cancer cell. Of course, we are going to mention now what's a proto-oncogene and what's an oncogene, but I like to present this diagram on how a normal cell can turn to a malignant cell. So by exposure to a physical damage, which can damage our DNA, a proto-oncogene can be activated and can turn the balance of the cell from normal to malignant. So what are the features of a cancer cell? A cancer cell replicates immortally. There's no control on its proliferation. It has many proliferative signals. It evades growth suppression. It's not controlled by tumor suppressor genes. It evades any growth, any control of growth. It can metastasize. There is increased angiogenesis in a cancer cell in general due to stimulation of the vascular endothelial growth factor, for example. A cancer cell resists cell death. And these are the main features which ensure that the cancer cell will replicate, will divide immortally without any control, and it will escape even the immune response. It has all these features which maintain its life, and it avoids, it becomes an immortal cell. This slide here, it's actually a very nice presentation and illustration of the molecular basis of a cancer, and it expresses the cancer genes. We can say that cancer can be acquired or inherited, but I want you to know that cancer is 80 to 90% an acquired disorder, an acquired somatic mutation. You can be exposed to chemical carcinogens, physical damage, biological carcinogens like viruses, for example. All these can lead to somatic mutations and lead to acquiring cancer or carcinogenesis initiation. Only 5 to 10% of, or almost 15% of cancer is inherited. And the main difference is that this mutation passes to the other generations because it's a germline mutation, but it only constitutes about 15% of the causes of cancer. Cancer can be due to replication. Acquired cancer can be due to replication errors. And I want you to know that replication errors are very rare. I know you studied replication, and you know the sequences of events taking place during replication and the DNA polymerase, and that the DNA polymerase is very fiddle, and it does not do any errors during replication due to its proofreading activity. And that we also have the guardian of the genome, which is the p53 gene, which prevents any error during replication and stimulate DNA repair if a damage took place. But in spite of all these proofreading activities, in spite of all these precautions and all these fidelity of replication, there can be some replication errors that can lead to cancer. These errors, they do not exceed 10 to the power -7 per cell per generation. So it's very rare to have replication errors, but they occur. So we cannot ignore that sometimes spontaneous replication errors can be the cause of cancer. Accumulation of mutations can lead to cancer. Or error during DNA repair, DNA damage of the DNA repair genes, for example. Or exposure to carcinogens like chemical carcinogens, physical damage, biological like oncoviruses, like we said before. So these can lead to alterations and mutations, which are of somatic cell mutations. On the other side, some inherited mutations which take place in germ lines, they include mutations or defective DNA repair genes, or they may affect tumor suppressor genes, or activation of oncogenes, for example. So all these mutations are result in carcinogenesis, and we cannot ignore the role of microRNA and epigenetics. Epigenetics and microRNA, they are not considered mutations. Mutation means change in the sequence of the DNA. So all these factors, they led to cancer because they changed the sequence of a DNA. So they resulted in activation of an oncogene, which is a growth promoting protein, or inactivation of a tumor suppressor gene, or alteration in the genes needed for apoptosis, or defective DNA repair genes. All these are mutations, whether they are acquired or inherited. But epigenetics and microRNA role here in carcinogenesis does not involve mutation. It's not a mutation. Epigenetics, it changes the DNA regulatory mechanism for gene expression. When we have an epigenetic alteration, there will be an effect on the rate of gene expression. For example, inhibition of the expression of a tumor suppressor gene, or excessive expression of an oncogene. So epigenetic here is not a mutation, it affects the gene expression, leading to uncontrolled cell growth. MicroRNAs also, we are going to explain them in some details. They cause cancer, they can be oncogenic because they affect the translation process, or they inhibit or hinder the stability of a messenger RNA, affecting the expression of some important tumor suppressor genes, for example. We are going to say all this in detail, but this is just an introductory to the main genes. These are the cancer genes we are going to talk about, the main genes which can be affected during carcinogenesis. I want you to know a very important information as well, that cancer cannot occur due to mutation in one of these genes only. No, it's not a single cell mutation. It's not a single gene mutation, to be precise. It's an accumulation of mutation. So in order that the cell, a normal cell, turns to a malignant cell, it requires accumulation of mutation, not just mutation of one gene. So we said that these are the genes which are going to be affected, the cancer genes. Now we are going to explain this part, the part talking about the DNA damage leading to mutation. We are going to explain this part now. So what are the causes of DNA genetic damage? These can be environmental carcinogens, like the radiant energy, exposure to UV, X-ray, gamma rays. All these are called, they can induce physical damage. They can damage our DNA. For example, the UV exposure to UV light, ultraviolet light can induce thymine dimer or TT dimer. We know that the DNA double helix is characterized by base complementarity, and that the T forms 2 hydrogen bonds with the adenine, and guanine forms 3 hydrogen bonds with cytosine. This is a basic knowledge. But when this DNA strand is exposed to a high dose of ultraviolet, let's say a high dose of ultraviolet light, and for long time, exposed to this wavelength for a long time, and especially when this person, for example, he doesn't have enough melanin, so the UV light will pass to the DNA and cause this damage, which is called the TD dimer or the thymine dimer. Like we have this adjacent cross-linking between 2 adjacent thymines. This, of course, is a kind of mutation. If it's not corrected, it's going to lead to a mutation, and it can lead to cancer. We can mention here that this type of mutation should be corrected by a special type of repair called the nucleotide excision repair. A set of enzymes has to come over here and remove this dimer and replace it with the normal bases and ensure normal base pairing and restore the normal double-stranded helix to stop this mutation from taking place. In case there is mutation in the genes responsible for the repair, here there is a mutation in the DNA repair genes, so this error will pass on to the next generation of cells, and it can lead to a type of cancer which is called xeroderma pigmentosum. It's a type of skin cancer. It's a type of skin cancer taking place. So can we avoid this background noise?
Excuse me.
Can you please mute your mic?
तो इसको दिक्कत होती है लगाने में। हम तो आपके घुसा देते हैं तो हमारा तो एंगल बना है, इसको ज्यादा तमीज से बोल के इसको निकालने में आसानी हो। तो जब ये वो फंसाता है ना,
am I audible? Can we please mute?
क्योंकि ये सोचते हैं कि मैं इस गेट से।
Okay.
Dr. Dalia, I have muted also. May you please unmute your mic?
So can you hear me now? Okay.
Yes. Okay.
Okay. So we were talking here about if in case there is a mutation in the DNA repair system, which is going to repair this error, in case there is a mutated DNA repair mechanism, this error will pass unnoticed to next generation, and it will cause some type of genetic disorder called xeroderma pigmentosum. in which these people are susceptible to skin cancers, especially that they are fair skin and they don't have enough melanin, because melanin will act as a protective barrier, a physical protective barrier, preventing much of this ultraviolet rays to induce the thymine dimer. So that was an example of an environmental carcinogen that can lead to DNA mutation. And I mentioned here a genetic disorder, which can be not due to this radiant energy, but the genetic mutation here is due to mutation in the genes responsible for the repair. Okay. So let's move back. So radiant energy, the main mechanism is prolonged exposure to UV sunlight, which is related to skin cancers, particularly if there is decreased melanin and disorders in DNA repair mechanisms. Also, the X-ray and gamma rays are mutagenic because, for example, X-ray was double strand breaks in the DNA, in the chromosomes, and gamma rays, both of them also, they result in excessive production of reactive oxygen species, which lead to DNA damage as well. So let's move to the chemical carcinogens. There are other categories of carcinogens, like the chemical carcinogens. Almost 80% of cancer causes are due to chemical carcinogens. They are due to environmental pollution. We are exposed to many cancer carcinogens all around us in the environment. They act mainly by modifying the DNA bases. They form covalent adducts with the DNA molecule. And depending on the extent of repair and damage, a variety of mutations can accumulate, leading to cancer. Like I just mentioned, that cancer is not a single event. It's not due to mutation of a single gene. No, it comes from accumulation, multiple accumulations of many genes involved in oncogenesis. Okay, so this is the mechanism by which the chemical carcinogen form the adducts I'm going to talk about. Chemical carcinogens, they form covalent cross-linking with the DNA bases. Because DNA is nucleophilic, it has electrons, it has negative charges, and the chemical carcinogen is an electrophilic, which lacks electron. So DNA forms like adducts with this chemical carcinogens, leading to mutation, and this mutation can be passed on with the DNA replication. It's an important point to note here is that these chemical carcinogens, they are not necessarily ready to bind to DNA. Sometimes they become more carcinogenic when they are metabolized by the liver. They are metabolized by cytochrome P450s to be more carcinogenic. They enter your body as pro-carcinogens, and they can turn to carcinogens by metabolism in the liver, some of them, okay? Other types of mutants are oncogenic viruses. Almost 15% of human cancers may be caused by viruses. Both DNA and RNA viruses can be carcinogenic. DNA viruses, they downregulate tumor suppressor genes, while RNA viruses, they carry oncogenes in their genomes. So a virus here causes carcinogenesis because it integrates its genetic material into your own genome. They can carry oncogenes in their genome, so they can transfer an oncogene into your genome, introduce an oncogene into your genome, or they can, like DNA viruses, they can downregulate a tumor suppressor gene. This is a diagram showing how an oncovirus, of course, the oncovirus, the virus enters your cell and it passes to your DNA. Either if it's an RNA virus, it's going to be converted to a complementary DNA by reverse transcriptase and to be incorporated into your genome, or it can be a DNA virus which can enter your genome directly. When a viral genome is integrated in the DNA, like I just said, that it can either carry an oncogene like an RNA virus, or it can introduce cause an inhibition of a tumor suppressor gene like DNA virus. These were the main causes or the main carcinogens which can cause genetic damage, environmental carcinogens. Now we have that some types of cancers are hereditary. Several familial conditions predispose to hereditary cancers, like mutations found in some specific genes. I mentioned one when I was talking about the TT dimer. We said that there can be genetic mutation of the enzymes responsible for the DNA repair mechanism. So they can be causes of some hereditary cancers. Mutations are found in specific genes, for example, tumor suppressor genes present in germ cells, so they are inherited. Appropriate genetic screening of families is always very important for early intervention. For example, if we are going to see here this table, it illustrates some types or some examples of hereditary cancers like familial retinoblastoma. Here, the mutant gene is a tumor suppressor gene, which is retinoblastoma gene. It's a gene which controls your cell cycle, and it prevents the progression, or to be more precise, it controls the progression of your cell cycle from one phase to another. So in case this gene, which regulates your cell cycle and prevents abnormal or excessive cell growth, if this gene is mutated, so the cell will control immortally, it will not be controlled, it will divide without any control. So this is one type of an inherited type of cancer. Familial adenomatous polyposis, the APC gene is also a tumor suppressor gene, and when this gene is mutated, it can cause some types of familial adenomatous polyposis. This cancer is of genetic inheritance, and thus early screening of these patients is very important for early therapeutic intervention. So that's why it's very important to know the basic knowledge. It's very important to know the genetic base of any disease, because early diagnosis will lead to early intervention and will lead to better outcome. Hereditary non-polyposis colorectal cancer is another example. It's an hereditary type of cancer, but here the mutation is in the mismatch repair genes. We said that the DNA can be damaged, but we have an efficient repair system. In case there is a mutation in this repair system, this can lead to types of hereditary cancers, like the xeroderma pigmentosum we mentioned, with the nucleotide excision repair failure mechanism. And here with the hereditary non-polyposis colon cancer, the defect here is in the mismatch repair system. Breast and ovarian cancers are very well-known examples of genetic or hereditary cancers. Like defects or mutations of BRCA1 and BRCA2 genes. These are tumor suppressor genes as well, and they are expressed in the breast. And when there is mutation of these genes, they can lead to hereditary cancers, like breast cancer and ovarian cancer. So for instance, if there is early diagnosis and early genetic analysis for such individuals who carry these genes in their families, who have inherited cancers of BRCA1 and BRCA2. If you are going to do early genetic analysis and genetic testing for the daughters, for example, you can allow for early intervention. Some will choose to do an early mastectomy or ovariectomy to prevent such sequelae. But of course, my own opinion, it's up to the patient, of course. It's something controversial, actually. So if we talk in more details about cancer genes, in the previous slides, I was discussing with you that we can have mutations in many types of genes, like oncogenes, tumor suppressor genes. I'm going to focus on these two parts. I'm going to explain them in detail. So we are going now to talk about the cancer genes, the genes that can predispose to cancer, how they are mutated by all these carcinogens. So mainly we have oncogenes are tumor suppressor genes. They are two main classes of cancer genes. They are very important to know. The oncogenes are derived from proto-oncogenes that normally stimulate cell growth. Now, normally we have proto-oncogenes. We can call them like they are like growth factors. They stimulate the cell to grow, to proliferate, to divide, to in order to replace the already dead cells to allow cell renewal and new cell formation. So proto-oncogenes are normally present in your cell to promote the normal cell growth. In case these proto-oncogenes are mutated, they can turn to oncogenes and they can lead to accelerated, uncontrolled cell division. So proto-oncogenes can be converted to oncogenes by one of the following: mutation, which we were talking about earlier, like chemical carcinogens or viruses or whatever. Promoter insertion. What's promoter insertion? That, of course, you know that in order to start the expression of a gene, you have a promoter which is upstream of the transcription site of the gene. And so this promoter helps the transcription process and help the RNA polymerase to bind and to start the transcription. So if you inserted a promoter, you're going to enhance the transcription more and more. Enhancer insertion, which will also stimulate the expression of this oncogene, chromosomal translocation, and gene amplification. Now let's look at this diagram, how a proto-oncogene can turn to an oncogene. It can turn to an oncogene if there is a mutation, point mutation. So it turns from a normal gene that promotes normal cell growth and cell division into a protein which is hyperactive, which stimulates uncontrolled cell division in case there is a point mutation. Gene amplification, that this gene is expressed uncontrollably, normal growth stimulating protein in excess. So this oncogene is expressed in excess. There is many of this oncogene protein, so it's going to stimulate more cell division. Translocation or transposition. Translocation or transposition, it means that here I introduced a new promoter to this oncogene, so it's going to be more expressed. Now let's look at this example. How can translocation or chromosomal translocation lead to activation of an oncogene? If you look at this example, it's a very well-known example, which is the Burkitt lymphoma example. There is here a translocation between chromosome 8 and chromosome 14. Now, normally chromosome 8 bears the MYC gene. What's the MYC? It's one of the oncogenes, okay? If you look over here, we're going to see that we have some types of oncogene. I placed some examples here, like the MYC, which promotes cell growth and increase cell cycle progression and DNA replication. It's mutated in a variety of tumors. So this is an oncogene promoting cell growth. So here, let's get back that chromosome 8 has the MYC gene, which is a normal proto-oncogene promoting the normal cell growth. And chromosome 14 has the gene for IgG, or immunoglobulin G, for expression of the immunoglobulin G. So in case there is a spontaneous translocation, chromosomal translocation between chromosome 8 and chromosome 14, part of chromosome 8 is going to be passed to chromosome 14, and part of chromosome 14 is going to be translocated to chromosome 8. Now we have part of chromosome 14 bearing the MYC gene. Now the MYC gene is inserted adjacent to the gene which expresses the IgG. So it's now inserted adjacent to the promoter of this gene. Normally the promoter of this gene increases the expression of IgG because it's an immune, it's an antibody. So we express it normally in the cell in excess. So in case this MYC gene is introduced near to the promoter of the IgG, it's going to be overexpressed. So now the translocation between these two chromosomes led to the following, that the MYC gene is now inserted near the promoter of the IgG. So the promoter of the IgG is going to stimulate the expression of the MYC, and now it's going to be an overexpression of the MYC protein. So it's going to turn to an oncogene, which was previously expressed in small amount to promote the normal cell growth. Now it's expressed in huge amount to increase and to make the cell dividing uncontrollably. So this is an example of how genetic alterations or gene changes, genetic changes can lead to activation of an oncogene. What about tumor suppressors? So tumor suppressors, they normally inhibit cell growth and cell division. But before we move to tumor suppressor genes, let's give some examples about these oncogenes. We just mentioned the activation of oncogene. Let's give some examples of oncogenes that we have. Let's say some criteria first, that oncogenes, we need to have mutation of the two alleles. One of the two alleles is sufficient to produce oncogenesis or to cause carcinogenesis. So normally we have from every gene two alleles. Mutation in one of them is enough to start or to initiate carcinogenesis. Gain of function of a protein that signals cell division, and an oncogene is normally a proto-oncogene. Now it gained additional function that increases and signal more and more cell division. So normally it was a stimulator for cell division. Now it gains another additional function that it increases or enhances more cell division. Mutation arises in somatic cells. It's normally not inherited, but it can be inserted by a virus, like we said before, the DNA viruses. They can, the RNA viruses, they can insert an oncogene. They perform some tissue preference. For example, the retinoblastoma here, the retinoblastoma gene, it regulates the cell cycle and it's responsible for retinoblastoma. Hence, it's called retinoblastoma gene because it's mainly expressed in the retina. So it represents some tissue preference, like we said before. No, this is tumor suppressor gene. I'm sorry. Okay, here it's I'm talking about the oncogene. No, we're not moving to the tumor suppressor gene right now. Okay, so some examples of oncogenes are the MYC, an oncogene that encodes a DNA binding factor that altered transcription, involved in cell growth, cell progression, and DNA replication, and it's mutated in a variety of tumors. This is an example of an oncogene, which is the MYC. Another example is the RAS family of oncogenes encoding for DTPs. So this family is mainly involved in signaling. And you are going to know that many of the causes of tumors is defective signaling or enhanced signaling mechanisms. So one of these is the RAS oncogene, which increases the GTPase activity. And in mutation of this RAS oncogene will lead to many types of cancers as well. These were examples of oncogenes. Now let's move to the tumor suppressor genes. They act completely the opposite of oncogenes. Oncogenes stimulate cell growth, while tumor suppressor genes, they inhibit cell growth and cell division. When such genes are altered by mutations, some of which are inherited, like the RB gene or the retinoblastoma gene, their inhibitory effect is lost if they are altered by mutation. So one is the oncogene, which stimulates cell growth, and when mutated, it will stimulate uncontrolled cell growth. And the other is tumor suppressor gene, which normally inhibits cell growth. So when it's mutated, the cell growth will go uncontrollably, okay? Here, both copies of the tumor suppressor gene must be mutated to lose the inhibitory effect on growth. So here the difference, here both alleles must be affected in order to lose the control over cell growth, okay? Examples of some tumor suppressor genes, they also often strong tissue, or they show some strong tissue preference, like the retinoblastoma and P53. Retinoblastoma is a tumor suppressor gene present in the retina, and that's why it was first discovered to cause retinoblastoma. and the P53, it's a tumor suppressor gene that is normally expressed in many cells, and it's mutated in almost 50% of human cancers. We have a mutated P53. P53 is mainly considered with cell cycle arrest if there is a mutation. Let's see this diagram. If you have a normal P53 and if a DNA damage took place, P53 normally acts in the late G2 phase and the early S phase of the cell cycle. So they detect any damage that can take place during replication, for example. So they stimulate some apoptotic proteins, some apoptotic genes, in case the error is not corrected. So first they check, they act as a checkpoint. Here, they act as the guardian of the genome. They check any error, they detect the error. They allow the cell to stop dividing, cell cycle arrest, to allow time for DNA repair. Then, if the DNA is repaired, well and good, and we can restart the cell cycle. If the DNA is not repaired, so we are going to move to apoptosis or programmed cell death. So here, P53, if there is no correction of the error, it will stimulate apoptotic genes to produce apoptotic proteins and stimulate programmed cell death. Okay, what if you have a mutated P53? Cell cycle will continue with the error. Now, here, let me remind you that we need to have mutation of the two alleles of the P53 in order to initiate carcinogenesis. So in case you have mutation of the two alleles, what will happen? The P53 will not be acting as the guardian of the genome. It will not detect the error. So the cell cycle will go on. The cell will divide, it will pass to the M phase and it will divide without any notice. So the cell will become cancerous. We can describe the P53 as the brakes. If you have a tumor suppressor gene which is active and it's healthy and it's normally expressed in both chromosomes, you are going to have the brakes active and they are going to stop any cell division with an error. But if you have no brakes at all, the cell is going to turn to malignant. Of course, adding to this, if you have another factor which is an active oncogene, like we said that the carcinogenesis is cumulative effect of many mutations. For a cancer, for a cell to turn to a cancer cell, you can have like 5 or 6 mutations, like 5 or 6 genetic mutations. So basically, it does not involve a certain gene mutation. You can have a tumor suppressor gene mutation and added to this an active oncogene or a proto-oncogene mutation. Of course, I would like to discuss all these details, but I know that there is a time limit, and it's a very huge subject. It has many knowledge. So I adopted this model in order to get to know more how accumulation of mutation can lead to cancer. This model was describing the colorectal cancer, described by Vogelstein, who described this model in 1990. It was actually the first genetic model to be described about the multi-step genetic mutations that take place in order to turn to normal epithelium of the colon to a cancer or to metastatic cancer, to carcinoma or metastatic cancer. As you can see, this diagram, it's a simplified one. It illustrates how a normal epithelium can turn first to a dysplastic epithelium by mutation of one of the tumor suppressor genes, which is the adenomatous polyposis colon gene. Then mutation of further accumulation of further mutation of the KRAS, which is an oncogene. It will turn to adenoma, more cell growth, and it will lead to turn to an adenoma. Additional mutation of a tumor suppressor gene like P53, it can turn to a carcinoma. Then it can turn to metastatic cancer. Of course, these are not the only genes here involved. There are many genes, and there is another diagram which can be more complicated, but I want to to go to the simple one in order to understand the basics, that accumulation of many mutations can lead to the progression of cancer from normal epithelium to dysplastic, which have some changes in the criteria of the cell itself, to adenoma, which is a tumor growth, or more multiplication of cells, to carcinoma, to metastatic cancer. This is one of the genetic inheritance genes responsible for the familial adenomatous polyposis. And addition of other mutations can lead to cancer. Normally, it's benign, it's not malignant, but it can turn to malignant with accumulations of more mutations. Also, let's see this one. Okay, cumulative alterations in the genome with progression, with progressive increase in size, dysplasia, and invasion. This is the slide the diagram I was describing, the model, the genetic model. Mutations, whether germline or somatic, in tumor suppressor genes and mismatch repair genes, inactivation of the APC, mismatch repair genes, methylation abnormalities, proto-oncogene mutation, for example, the KRAS, additional mutation of a tumor suppressor gene, for example, the P53. All these are the sequence of events which can take place to lead to accumulation of mutation and to turn the cell from normal epithelium to adenoma, to carcinoma, to metastatic carcinoma. So this is just an enumeration of the mutations which can take place in the colorectal cancer model. A large tumor may contain a variety. Now this is important. A large tumor may contain a variety of cells with different genotypes, which makes successful treatment more complex. What does this mean? It means that this tumor, if we look at this tumor, if you examine each cell, you can see some genetic alterations present in one cell and not present in the other. We can see that these are not having the same genotype. They can have different genotypes. So this is part of particular importance when you choose the line of treatment, when you study why some patients have resistance to chemotherapeutics, because when you give a single chemotherapeutic agent, you may be targeting one type of mutation. Maybe you need to add more specific chemotherapeutic agents to target the specific mutation or many mutations involved in this model. So it's very important to study and to know the genotypes of a cancer cell, the different genotypes of a cancer cell, like we see here, there are multiple mutations. You are not targeting a single mutation. You have to know this in detail. You have to study the cell, and here come the single cell sequencing, a methodology or a tool by which you can study the genes of a specific cancer cell for a specific individual. So you can be aware of all the genetic damages that took place in this cell, which can be different from the neighboring cell, which can be different from another individual having the same type of tumor. So that's why it's very important to dig into the molecular staging of the cancer cell in order to choose the appropriate treatment, which will provide a better outcome for the patient. MicroRNAs, now we studied the cancer genes. We mentioned cancer genes and some mutations of some cancer genes. We can talk about the microRNAs, as they are key players now in carcinogenesis and tumor metastasis. It's not only about the basic genes of the DNA. No, some regulatory genes, they play a role in carcinogenesis and tumor metastasis as well. MicroRNAs are one example of these key players. Let's introduce microRNA. I'm sure you know what's microRNA, but let's talk in brief. MicroRNAs are normally one of the non-coding small nuclear RNAs, which are non-coding. They do not code for proteins, but they are mainly present for regulation of gene expression. MicroRNAs are produced from their genes on the DNA by polymerase II enzyme, RNA polymerase II. They are transcribed, and they have a 5' cap and 3' polyadenated tail and so on. They are processed by many nucleases. Some of them are present in the nucleus, the drosha, and they are exported through the nuclear membrane to the cytoplasm, where further processing will take place through the dicer protease in order to produce later on the mature form of the microRNA. One of the two strands is now chosen, and this will be the functional mature microRNA that will act on its target RNA. Every microRNA has a specific sequence. It ranges from, it's a small molecule, small single-stranded molecule from 19 to 22 nucleotides. It targets a specific sequence on the 3' non-coding region of the DNA, of the RNA, of the messenger RNA. And by this binding, the microRNA can either stop translation, inhibit translation. So this is a form of regulation of gene expression. The messenger RNA is already transcribed, but now comes the microRNAs to prevent, to stop the translation process. So it's a form of regulation of gene expression. Another type of regulation that it can alter the stability or destabilize the messenger RNA, degrade the messenger RNA, so also no translation will take place and no protein synthesis will take place. An unusual binding that it can bind to the 5' end of the messenger, enhancing translation, turning a quiescent cell present in the G node phase to a proliferative cell, producing more proteins. So the microRNA, it normally regulates gene expression by inhibiting the gene expression, by inhibiting the translation process. And we have, normally, we have microRNAs, which can be oncogenic, which can turn the cell to a malignant cell. And we have other microRNAs, which can be tumor suppressors. For example, they can degrade here an oncogene. So it depends on the site of binding. It depends on which microRNA, which is going to be the target of the microRNA. If the microRNA targeted a messenger RNA for a tumor suppressor gene, so it's going to turn the cell to a carcinogenic cell or a cancer cell. If the microRNA targeted messenger RNA of an oncogene, it's going to prevent cancer. So we have both. We have oncogenic microRNAs and we have tumor suppressor microRNAs. This is normal. What if we have aberrant or mutation of this microRNA? What will happen? What will happen is that we can have excessive expression of an oncogenic microRNA, like microRNA 21 or 155. Some of them are involved in cancer breast and cancer lung, and they already passed through a lot of researches now that we can use this microRNA actually as diagnostic biomarkers. They are under research, extensive research, and some of them can be underexpressed under expression of a tumor suppressor microRNA. We can detect an underexpression of a tumor suppressor RNA in some cancers, like microRNA 16 and LET7. So we have oncogenic and we have tumor suppressors, and any of them can lead to carcinogenesis, because here there's overexpression of the oncogenic microRNA, and here there is underexpression of the tumor suppressor microRNA. So this means that microRNA sequences can be mutated as well, and thus they can lead to dysregulation of gene expression and proceed to carcinogenesis as well. Recently, they are being investigated, like I said, as diagnosed or prognosed markers, and some of them are currently being used in clinical trials as therapeutic targets. Clinical trials are going on right now, and they are trying to use this microRNA as therapeutic targets. For example, you can design an oligonucleotide which can bind to the microRNA, so it binds to an oncogenic microRNA and prevented from causing oncogenesis, and thus potentiating the response to anti-cancer chemotherapy. So now we can design some oligonucleotide sequences that can bind inside the cell to the oncogenic microRNA, preventing it from its action and preventing it from causing further progression of the tumor, and thus they can be conducted as anti-cancer chemotherapeutic. Of course, all these are still under trials, under research, but there are promising results indicating the importance of studying such molecules. Okay. Now I want to move to this part. Okay. Extracellular vesicles and cancer, or let's say this part first, that cancer cell evade apoptosis. A cancer cell as well, a cancer cell bypasses the phase of apoptosis. A cancer cell has mutated tumor suppressor genes, has mutated apoptotic proteins. So normally, if you have an unfixable DNA damage, mutation of apoptotic genes or overexpression of an anti-apoptotic gene will lead that the cell continues dividing, and this can lead to cancer if you have a mutation of the apoptotic genes. But if you have the apoptotic genes functioning well, like the BAX, the P53, the P53 will upregulate this apoptotic gene and it will enhance apoptosis. So normally we have apoptotic genes like P53 and BAX, which will allow the cell to go to apoptosis in case there is an unfixable DNA damage, and we have an anti-apoptotic gene like the BL2, which is an anti-apoptotic in order to prevent excessive apoptosis. This is the normal, that we have apoptotic and anti-apoptotic. The apoptotic will only act if there is an unfixable DNA damage detected by P53. What if you have mutation of these apoptotic genes and upregulation of the anti-apoptotic? The cell will continue to divide with the unfixable DNA damage, and this can lead to cancer. So this is one of the criteria of the cancer cell, that it bypasses apoptosis. It's immortal. Okay. Now, another an exciting era of oncology is the discovery of the extracellular vesicles and their relationship to cancers. What are the extracellular vesicles? They are the exosomes or the microvesicles that are normally released by normal or diseased cells. So you can have a normal cell or a diseased cell releasing these exosomes. These exosomes are for intercellular communication. The exosomes can carry proteins to a neighboring cell. Now we discovered that they carry genetic material. These exosomes, they are surrounded by a lipid bilayer, and they can be released by a normal or a cancer cell. So let's imagine if the cancer cell released an extracellular vesicle containing non-coding genes or non-coding molecular molecules like RNA, microRNAs, or non-coding RNAs, or circular RNAs. All of these can be loaded into these vesicles. What if this cell is malignant and it's releasing these microvesicles containing the genetic material? So this genetic material will definitely affect the surrounding cells or the distant organs. So if you see here this model or this diagram, this figure shows that the donor cell, which releases now an extracellular vesicle or microvesicle, maybe this cell is a malignant cell. This microvesicle will be carried to another cell. It can bind by membrane fusion to another normal cell or by endocytosis, and it can transfer the genetic material. This genetic material, which can be a microRNA, can be transferred to another cell and affect the gene expression in this normal cell. So this is one way by which metastasis can take place, and this is a very important discovery, that we discovered that the intercellular communication by exosomes These exosomes may be responsible for metastasis because they carry genetic material. And this genetic material, like microRNA, it affects gene expression. So this is one way by which these extracellular vesicles can cause metastasis or angiogenesis or tumorigenesis, depending on the genetic material transfer, depending on the effect of this genetic material on the DNA of the new receiver cell. We can use these extracellular vesicles actually in the lab. You can use it because it's excreted in the patient's saliva, urine, blood. All these are under investigations now that we can use them as biomarkers to diagnose some types of cancers or prognostic signs, to detect these extracellular vesicles in the saliva or blood or urine of the patient. So they are very stable and they transfer microRNA. They can be used also, but they can be used as delivery system, therapeutic agents. For example, we can load these extracellular vesicles, we can load them with a specific microRNA or anti-microRNA to treat a disease. We said before that microRNA can be tumor suppressor or oncogenic. So what if we load these extracellular vesicles with a microRNA that is tumor suppressor? It can help to treat some diseases. It can help to treat the progression or improve the response to chemotherapy. All these, I just want to emphasize that all these are under research and all these are under trial. They're not used as therapeutics yet, or maybe they are used in narrow spectrum, I don't know about, but they are still used under, they are still under research. But they hold promise. Now, we talked about the microRNA. Let's revise. We talked about the cancer genes, the oncogenes, and the tumor suppressor genes in short, of course. And we talked about how the cancer cell evade apoptosis. We talked about the microRNAs and the extracellular vesicle. Now we are going to talk about another factor responsible for carcinogenesis, which are the epigenetic changes. Epigenetic changes are involved in cancer. And let me emphasize that epigenetic changes can be inherited or they can be caused by environmental hazards. The environment around us can affect our genes. It can cause epigenetic alterations. It can affect our gene expression. So what's meant by epigenetic? Epigenetic, it means all the studies that are associated with mainly the proteins or the chromatin which forms around which the DNA is bound. It describes the post-translational epigenetic changes, mainly talk about the post-translational modification that we can do to histones. What are histones? Histones are the proteins around which our DNA is tightly bound in the chromatin structure. Our DNA is not naked. It's tightly bound around histones, histone proteins, to form the chromatin. Now, this chromatin can be modulated. It can be modified. Normally, this is the chromatin. Normally, this is the DNA in purple wrapped around the histone proteins, which is in brown. And if we change the chemical groups on these histones, the DNA can be more tightly bound, so there is no gene expression, or it can be relaxed, and so this will increase gene expression. So by modifying chromatin, by modifying the chemical groups on the histones, we can modify our gene expression, the DNA expression, and thus this will definitely play a role in cancer. So this epigenetic changes, we can modify histones, like I said, we can modify it by methylation, by acetylation or deacetylation, by phosphorylation, many mechanisms. For instance, I'm going to mention one example. If we modify these histones, for example, by acetylation, we're going to increase the gene expression. If we are going to modify the DNA sequence itself by methylation, we are going to inhibit the gene expression. Generally speaking, a hypermethylated DNA is not expressed, is not actively transcribed. So now they used this methyltransferase inhibitors in order to treat some types of leukemias and lymphomas. How come that they use some drugs to inhibit the excessive methylation? What will the excessive methylation do to a DNA cells? It will inhibit gene expression. So in some type of cancer, you have a hypermethylated DNA, a hypermethylated promoter of a certain gene. This hypermethylated promoter can lead to underexpression of a tumor suppressor gene. So if you have a hypermethylated tumor suppressor gene, it's going to be inactive. So it's going to stimulate carcinogenesis. It's going to be one of the factors inducing carcinogenesis. If we took some drugs to inhibit this methylation process, this can be used in treatment of some types of cancers, and they tried it with some types of leukemias and lymphomas. How much time do we have?
As much as you need, definitely.
Thank you. So what time is it? Can I know?
It's 7:03, so we have too much time to. So we have 2 hours have lost.
2 hours?
No, sorry, 1 hour have lost. Just 1 hour have lost.
We just passed through 1 hour, right? Okay.
Yes. Yes.
Okay. All right. That's fine. So let's get back to the epigenetics. Normally, that we have our genes and we have epigenetic changes. So in order to maintain our genome stability, we have some modifications of the histones. Let's see this one. In order to maintain the genome stability, we have our histones acetylated in some parts. So these parts are actively expressed, and we have the histones deacetylated in some parts. So we have this parts constitute heterochromatin, or it's not expressed. So normally we have in our genes some genes that are actively expressed, always expressed, so their histones are acetylated, and we have some parts of the genome which is normally not expressed, and so the histones are deacetylated. So these are the epigenetic, normal epigenetics of the DNA, that some parts, some genes are normally expressed and some genes are very slowly expressed. So if there is any genetic alteration, can lead to cancer as well. But here, this epigenetic alteration, like I said, is not a mutation. It's a DNA methylation, histone modification, or by regulation through the non-coding microRNA we explained before. All these are called epigenetic. So epigenetic changes that affect gene expression can lead to cancer. Normally, we have some genes that are not methylated, that are methylated. So they are not expressed. What if these genes become suddenly expressed, like oncogenes or so the cell will be malignant? Normally, we have some genes like tumor suppressor genes, which are expressed at a certain rate. What happens if these genes their promoter become methylated? They will not be expressed, and thus the cell will turn to malignant as well. So epigenetic changes as well can lead to cancer, but they are not mutations. Mutations means that I change the sequence of the DNA by point amplification of an oncogene, like we explained before. So the cell can turn malignant cell if there is alteration of the epigenetic enzymes, or there is mutations that took place to the DNA sequence itself. Okay, so we want to emphasize now that epigenetics as well, which can be methylation of the DNA itself, methylation of the CG islands present in the promoter sequence of any gene, or histone modifications. All these is acetylation or deacetylation. All these changes may activate an oncogene or underexpress a tumor suppressor gene, leading to cancer. Okay, So normally we have the epigenetics controlling that some genes are constitutively expressed and some genes are not expressed. If there is change of these epigenetic, there will be expression, overexpression of an oncogene or underexpression of a tumor suppressor gene. Genome instability and aneuploidy are important characteristics of a cancer cell as well. What does this mean? What's a genomic instability? What's an aneuploidy? A microsatellite instability is one example of the genomic instability. Normally, we have a repeated sequence of DNA, short repeats and long repeats. We have our DNA is redundant. Our DNA is not only is not only formed from coding sequences. Some of the DNA, or the majority of our DNA, the majority of our genome is non-coding because it's formed from non-coding repetitive sequences, and we call them the short interspersed sequence or the long interspersed sequence. Microsatellite repeats. We have some repeats of the DNA like this model, the CCAA. This is a microsatellite repeat. This is present normally pattern, a normal pattern in our DNA. What if this pattern is disturbed? It can lead to cancer as well. So even disturbance, some disturbance of our non-coding sequences can lead to cancer. For instance, if we have here this DNA replication microsatellite repeat, there is an insertion of an excess CA. It's not normally present. Now there is an excess CA in the microsatellite sequence. If this error is repaired by the mismatch repair system, which is intact, so that it will be repaired and there will be no instability, the genome is preserved, the microsatellite sequences are now stable. What if there is failure of the repair mechanism, like we see in the mismatch repair deficiency? If there is a deficiency in the mismatch repair here, so this replication error will pass to the next generation of cells, and there will be a microsatellite instability. What's meant by microsatellite is that this sequence is extended. This sequence, for example, let me say a very, very modest example. For example, if this sequence is formed from 7 nucleotides, 7 repeats, now they are 10. This is called an instability because it affects the stability of the genome. Okay, now the number of the microsatellite repeats is amplified for no reason. So this may indicate that there is a mismatch repair defect in other important genes, which can predispose to cancer, because DNA mismatch repair deficiency will not only affect the microsatellite repeat, but it can be also an indicator that there is a mismatch repair deficiency elsewhere in important genes. So if you have a genome instability due to excessive or addition or extension of the microsatellite repeat sequence, this indicates that there is a DNA mismatch repair defect. And this is an inherited type of mutation, and it was detected in Lynch syndrome, which is a type of familial non-polyposis colon cancer, where there is extension or overexpression of the microsatellite repeats due to replication and mismatch, due to mismatch repair defect. This is a type of genome instability which takes place with cancer. So some types of cancer have genome instability, which means that the repetitive sequences can be extended. They are not stable. So in your DNA, even in the repetitive sequence, it means that your genome is not stable. It means that your repair system is not functioning well. It means that the repair replication error passed unnoticed. Okay? Some drugs are now being targeted to redirect or to affect this mutation or to correct this mutation. What about the chromosomal instability? Chromosomal instability is one type of aberrations taking place in a cancer cell as well. That you can see that, as you can see here, that a cancer cell multiplies very rapidly. The cell cycle is very short. and this can lead to telomere shortening, progressive telomere shortening. And this is a common feature of solid tumor, that you can have the chromosomal number is not exactly the multiple of the haploid, which is a poor prognostic sign. How does this happen? How does this happen? That progressive telomere shortening due to accelerated cell division, our telomeres, they get shortened very rapid. And this progressive telomere shortening is a bad sign that the cell is a cancerous cell. Now, this progressive shortening can have some hazardous effect on our chromosome, because it can lead to breakage of the ends of the chromosome and chromosomal cross-linking or fusions, and these are one of the chromosomal aberrations or the chromosomal instability that we can find in a cancer cell. So the chromosome can fuse, they can break, they can have a different number. For example, in a haploid cell, you have 23 chromosomes. In a diploid, it's 46. So you can have, it's not 46, it's 48, it's 50, whatever. So it's not the double of the haploid number. It's not the diploid number of chromosome, because now the chromosomes, there is critical shortening of the chromosome, and this allows chromosomal fusion and chromosomal breakage, and this is called aneuploidy or chromosomal instability. So the number of chromosomes is not exactly double the haploid number. So this will lead as well to telomerase activation. So this progressive shortening of the telomere in a very short time will, and this sequelae will lead to telomerase activation, and thus the cell will now become more and more immortal cells. So this will activate our telomerase. These progressive telomere shortening due to the excessive rapid division of cancer cell will stimulate the telomerase activity, and this will lead to more progression of the tumor. It can progress like the carcinoma to an invasive carcinoma. So that's why telomerase are being investigated, or they are attractive targets for some chemotherapeutics, because if you target the telomerase, you are going to stop the cell from the dividing. The cell will go to apoptosis, and it will not have any chance to divide anymore. Now we talked about cancer genes. I know that the lecture has many information and it may be somehow difficult, but we went through the cancer genes, we went through the genetic defects that can lead to activation of these cancer genes. Now, then we mentioned the role of the microRNA in carcinogenesis, the role of epigenetic changes in carcinogenesis, and now we are going to talk about the tumor microenvironment. How does it play a critical role in cancer development, metastasis, and response to treatment? If you see a cancer cell, you're going to, shortly, we're not going to go through all the details of this diagram or this figure, but as you can see here, that a cancer cell is full of immune cells. It has many immune cells. It has many growth factors. It has many signaling molecules. So how comes that a tumor scene immune cells, and these immune cells will not go to eradicate this cancer cell? Now, after asking this question, we have to know that the anti-tumor effect of immune cells is inhibited in response to tumor-derived signals. As you can see here, some scientists, by the way, believe that cancer is mainly due to, or a big part of the cancer progression was signaling by the oncogenes, signaling the cell to grow, signaling the cell to multiply, signaling angiogenesis, by all the growth factors, like epidermal growth factor, endothelial growth factor. So this cell is very active and it's very actively dividing. And so all these signals will definitely affect and will eradicate the immune system. It will prevent the immune system or the immune cells from exerting their anti-tumor effect. In fact, the T lymphocytes and macrophages, they become reprogrammed by one of the tumor promoting factors, which is the nuclear factor kappa beta, which activates or which changes or reprogram the T and the lymphocytes and the macrophages so that they become promoting more and more the tumor growth and survival. Thus, cancer cell escape the immune surveillance or escape the immune response, because as you see, the cancer cell is full of growth factors, signaling molecules. It definitely affects the T and the B lymphocytes through the nuclear factor kappa beta, so it reprograms the immune system and it escapes the immune response. Genetic analysis of tumor cells provided us with new insights into cancer. Now, all what I have mentioned in this lecture, it means that now we have new insight. We become more insightful of a cancer cell. We know all the criteria, not all of what, we know many things we didn't know before about the genetics of a cancer cell. So this will definitely help the diagnosis, help to follow up the patient, help the treatment strategies that we can use. The progression of the biotechnological tools in the late last decades, like the DNA sequencing, bioinformatics, the next generation sequencing. Now we can know the sequence of many tumors. This provided us with a comprehensive catalog of specific gene mutations. Now, this valuable revolution of knowledge allowed us to develop new diagnostic markers and develop the tailored therapy that each patient should be treated separately. Now we know that cancer is accumulation of mutation, and that each cell can be different from the neighboring cell in its genotype. So this means that we are not fighting a single mutation. When we give a chemotherapy, we are targeting multiple mutations. That's why we have to go through the single cell sequencing to create the multi-model treatment strategies that we can use. For instance, by all this knowledge, we could move to the targeted therapy or the immunotherapy, where you can use a monoclonal antibody to target a cancer gene which is expressed on the cell. For example, targeting the HER2 receptors in cancer breast. If we don't have a knowledge about the HER2 receptor and how it's mutated in cancer breast and how it's responsible for the aggressiveness of some type of cancer breast, we will never have known the targeted therapy which was giving monoclonal antibodies to target this HER2 receptor and to improve the outcome in this aggressive type of cancer, for example. So using the genome modulating technology like the gene silencing or genome editing like CRISPR, all these are important tools to identify genes that can cause or can accelerate cancer. And if you identify these genes, you can modulate your therapy, you can modulate your treatment strategy, you can use a multi-modal treatment, not using a single treatment which can result in drug resistance. So ultimately, personalized therapy for cancer in individual patients so that every patient has his tailored personalized therapy based on the genetic knowledge of this tumor in this particular patient. Now, this is what's called the precision oncology, and this is particularly important to all oncologists. Now, this is the same. I was just explaining that the fundamental biochemical and genetic mechanism involved in carcinogenesis, they paved the way to discover new and better therapy, and that studying this mechanism by cancer drugs develop resistance. We can currently consider this a very important topic for research. So we go to the molecular profiling of cancer in an individual patient to allow the oncologist to choose the most appropriate modality that targets the molecular abnormality in each tumor, or the personalized, what's called the personalized anti-cancer therapy. Such approach can significantly improve the clinical response to drugs and consequently increase the survival in certain cancers. And thank you very much for listening. And I hope you enjoyed the lecture and you get some benefit from the information. Thank you very much.
Definitely.
Thank you so
Thank
much.
you for
Thank you, ma'am.
I would like to personally
ma'am.
thank you for your presentation. We are very grateful for the time and talk to share your knowledge and experience about the molecular basis of oncogenes and so forth. So we truly enjoyed. So best of luck and thanks one more time.
My pleasure.
So dear students, if you have any kind of questions related to this topic, we would love to try to solve your questions. So we are glad to hear if you have any questions. Okay.
Sir, I have one question.
Yeah.
Sir, can you explain how the telomere phase is affected by the cancer cells? Like how the structure is decreased in size when they're getting affected by these cancer cells?
So if I heard correctly, you want to know how drugs targeting telomerases can decrease the size of the tumor? Is this your question?
Yes, ma'am. Yes,
Okay.
ma'am. That would be great.
So, okay. So as you know that a telomerase, you have a basic knowledge about the telomerase enzyme? How
Yes,
does it
ma'am.
act?
Little bit I know.
Okay, so we know that the telomerase is an enzyme which replaces the shortened ends of the chromosomes which happen with each cell cycle, right? So our chromosomal ends, they get shorter with each cell cycle, with each replication. And telomerases, they replace our enzymes which have reverse transcriptases to replace the shortened end of the chromosome.
Okay.
So in cancer, you have this telomerase upregulated, and you have this enzyme acting indefinitely. It avoids senescence of the cell. It prevents the senescence of the cell. The cell will not stop dividing. Okay? So what if you target this telomerase? What if a drug which will bind to this telomerase and inhibit its action? The cell will not be able to divide. The cell will not be able to go immortal. It will reach a stage where the chromosomes reach a critical end and the cell, it's not viable. The cell cannot survive anymore. So the cell will die. So this is one of the treatment strategies. It's not the only one. Like I said, that you need to have a multi-model therapeutic strategy. It's not only one drug. You don't give. We stopped giving one drug for cancer patients now. So it's one of the hopes that we can decrease the size of the tumor by preventing the cell division. So we can give anti-telomerase. Some drugs target the topoisomerase enzyme, which is an enzyme involved in replication. Some drugs target, they bind to the major groove of the DNA molecule itself. Some anti-cancer drugs, so they prevent the DNA from replicating. If you can give a drug which inhibits replication or which inhibits cell division, you will definitely affect the size of the tumor somehow. Okay?
Right. Thank you, ma'am.
You're welcome.
So.
Thank you, Ms. Dalia, once again for the very, very interesting presentation, very interesting lecture. I will just repeat, is there any other question from the student side? Otherwise, we can finalize today's meeting. Dear students, are there any questions?
No, ma'am.
So how seems everything was very clear and
I hope so.
Perfectly
Hopefully.
explained.
Hopefully.
Hopefully, yeah, perfectly explained. So we are so grateful. And thanks for your attendance, dear students.
Very pleased to be with you here. Thank you all. It's my pleasure to meet you and hope to meet soon again, inshallah.
Excuse me, I want to say something. Please, can you send us the PPT?
Yeah.
The presentation?
Yeah, sure.
Can you send it to us?
Yes, definitely.
Okay. Okay. Thank you.
You're welcome.
So the question is if this lecture is recorded or not, and the answer is definitely yes, which will be shared after the lecture for sure. So if we have any other questions, that's great. Otherwise, I think we're gonna finish, and this is the end of the story. I think.
Thank you once again very much.
Okay. Thank you, Doctor.
For the collaboration in the future. Have a nice evening. And dear students, thank you for attending.
Have a nice evening, all of you. Thank you. Bye-bye.
Thank you.
Bye-bye.
Thank you.